Good morning, everyone. I hope everyone is now suitably caffeinated. Right, so I'm going to present today on behalf of myself and Benita, who's in the audience, the latest data we have describing this first-in-class siRNA delivery path. So disclosures before we go any further, I'm an inventor listed on 2 patents covered in this work, and Benita, who is the CSO and CEO of Vitaka Therapeutics, has entered an evaluation and an option to license agreement between her company and GUEL at the university. So we heard yesterday about compartmentalization. I'm standing before you doing mutually exclusive tasks, believe it or not. I'm digesting my lovely breakfast whilst attempting to enlighten you with regards to this particular technology. So in order to do this, I separate the catabolic processes in my stomach from the anabolic and persistent processes going on in my cranial vault. What would happen if I decompartmentalized? Well, I would make my point, if only very, very briefly, that compartmentalization is quite an important physiological thing. It may give you a bit of a giggle, but I do assure you this talk will end fairly quickly. Cells do exactly the same thing. They have a lot of persistence and anabolism going on within the nucleus, the equivalent of the cell's cranial vault, and catabolic activities, digestion, going on in the cell's stomach or endolysosome. So the endolysosome is part of the late endocytic compartment, which is composed of lysosomes, the endolysosome, which is a hybrid between the lysosome and the late endosome itself. And the endolysosome is where digestion occurs. And you can see from a survival and physiological point of view why the cell would want to compartmentalize all of those catabolic processes. And it does that by keeping big molecules that can't readily partition through membranes inside the lumen of these compartments. And you can see this in the micrograph on the bottom right. These are squamous epithelial cells. You can see the negative staining kind of way where the nucleus is by the absence of vesicles. And you can also see vesicles that I've immunostained for the protein LAMP, lysosome-associated membrane protein, which is a very well used and very well known marker for the late endocytic compartment. I've also given these cells breakfast in the form of fluorescently labeled wheat germ agglutinin. And this wheat germ agglutinin has a fluorophore called Texas red associated with it. And I've pulsed the cells for 4 hours. I've given the cells a little wash. I've gotten rid of any non-internalized wheat germ agglutinin. And then these cells have been chased for a further 20 hours to make sure that all of this material is now within the late endocytic compartment. And indeed, you can see co-localization denoted by the yellow color in these squamous epithelial cells. Why am I so concerned with compartmentalization? Well, I'm also very interested in delivering large molecules to cells. Cas9, guide RNA, siRNA, antisense. None of these things can get past membranes, these phospholipid barriers, without help. There are leaky fusion events that go on within a cell, which is where you get misfusion between endocytic structures, and this accounts for very, very low, inefficient delivery. However, within the cell, there are processes that I think that we can exploit in order to improve our prospects for the delivery of large, non-membrane pervious materials. Because as we can see from this diagram, A, that is the outside of the cell, and material within these membrane delimited endocytic structures is not equivalent to B. Okay, so how on earth are we going to do this? I've always been a bit of a space geek. I'm sure that won't come as a massive surprise. And making reference to the Apollo moonshot, we have a repurposed intercontinental ballistic missile with a nuclear warhead on. And the warhead from that missile was removed and replaced with a life support system, which in the late 60s enabled us to go to the moon and back. Hooray, what a wonderful thing. In nature, we have protein toxins that have evolved. We're able to also remove and decommission the warheads from these toxins, leaving the rocket motors, which can be used and indeed have evolved to access this nuclear cytosolic compartment of the cell with relative high efficiency. There are a number of toxins that do this: ricin toxin, sugar toxin, cholera toxin, diphtheria toxin, to name a few. All of these have fairly scary names, even after, and you know, the stigma surrounding the names of these toxins remains even after you've removed the warhead. So we want to use one of these toxins. We want to detoxify it by removing the warhead, and we want to modify it so that it can carry a nucleic acid payload. And we've done that by taking a toxin component, removing the warhead, and replacing it with a nucleic acid binding domain. So we have two components to our delivery system, which is a protein called PA83, which oligomerizes to form a pore, not in the limiting membrane of the endosome or lysosome, but within an intraluminal vesicle, which you can see with this little blue mushroom in it in the cartoon, which is representing this oligomeric non-toxic PA protein. We also have the PA entry sequence called LF, or LFN in this instance, which has been fused in frame recombinantly to a nucleic acid binding domain. So we now have a system that can bind nucleic acids like siRNA, or we can swap out one binding domain for another and bind short sequences of DNA too. And we published that, yes, this indeed can deliver nucleic acids with high efficiency and low toxicity. We can make these materials using E. coli. Recently had the Vice Chancellor of the university being rather worried that I had Bacillus anthracis in the lab. I had to assure her that we didn't. And E. coli does a brilliant job of actually making these proteins. The proteins themselves aren't toxic and perform reasonably well. However, there is a bit of a caveat. PA is published when it forms a pore to have a very small hole in the middle, known as the phenylalanine clamp, which shouldn't be big enough to let nucleic acids through. But hold on, I've just told you that I actually have, well, we have published that this system works with high efficiency and low toxicity. How can that be if the phenylalanine clamp is too small, excuse me, to let a molecule like RNA through? Well, okay, we were wondering, first of all, have we made recombinant PA? Does it behave like wild type PA? And we oligomerized PA80 or PA63, which is the active form of PA83, took it up to the Rutherford Appleton laboratory and fired neutrons at it, as you do. The result of this was these experiments describing an annular structure that agreed in terms of its dimensions with the published PDB crystal structures. Okay, so the proteins seem to be behaving okay. Perhaps we're not actually sending material down through the middle of the toxin, perhaps like colicin or another molecule like that, we're going down the outside. Oh, that's rather peculiar. Or perhaps we're just bursting the endosome, as we're stressing it by having another protein that has hydrophobic domains that can span membranes. So first of all, we thought, let's have a look at this. Perhaps we can test these hypotheses and find out, perhaps with a little more clarity, what's actually going on. So Paul Luzio in Cambridge, a number of years ago, published a lovely paper exploring the specificity of vesicle fusion during endocytosis, where he loaded cells with hypertonic sucrose, which caused late endocytic vesicles to expand and swell. This has the effect of preventing intraluminal vesicle formation. Remember these ILVs, these intraluminal vesicles, they were the ones with the little mushroom on from the previous slide. And these are the structures that PA inserts into. The translocation event happens, loading the lumen of these ILVs, which then undergo back fusion with the limiting membrane of the late endosome, releasing cargo into the cytosol. So by loading up late endosomes with sucrose, we'll cause them to swell, preventing ILV formation. We thought the same thing would happen if we used wheat germ agglutinin, but at a slightly higher concentration, as wheat germ agglutinin will cross-link membranes, stabilizing them further, and also preventing the invagination of membrane necessary for ILV formation and efficient PA activity, i.e. translocation into the cytosol. So we thought we need some assays to test this. The first one we chose used the translocation of elephant diphtheria toxin A chain. Diphtheria toxin A chain is very, very toxic. It'll kill your cells very quickly if it can get into the cytosol. If it can't, as you can see from the graph in the middle, we don't actually hit an IC50. LFN DTA on its own doesn't actually cause a great deal of toxicity. That story changes quite dramatically if you add 5 micrograms per mil of protective antigen, at which point you have toxicity here in the, well, 50 to 100 nanogram per mil DTA concentration range. So you can see a characterization of DTA here, and this indeed is an assay that can measure the translocation of material over the PA pore. And we also know from the literature that if you RNAi down a protein like ALEX, which is critical for ILB formation, you reduce PA activity by about 80%. So yes, ILVs are indeed necessary for PA activity. We have a second assay where we're using HEC293 cells that constitutively express GFP fused to LAC-Z. LAC-Z will hydrolyze beta-galactosidase, and we can spectrophotometrically measure beta-galactosidase. Beta-gal hydrolysis in XGAL, XGAL is the substrate that turns blue. We can measure the amount of blue as an indicator of enzyme activity. If we introduce an antisense oligonucleotide into these cells via the endopore system, that is the PA and LFN-NBD nucleic acid binding domain, you get a reduction in the amount of blue material appearing, which is because you've just inhibited the production, or you've knocked down the beta-galactosidase gene. So we did that as well, and when we looked at siRNA activity in the presence of wheat germ agglutinin, you can see concentration-dependent inhibition of siRNA activity. You can also see wheat germ agglutinin concentration-dependent inhibition of LFN-DTA lethality. What is that telling us? It's telling us that wheat germ agglutinin, which is cross-linking membrane, is inhibiting PA's ability to function as a translocase. Similarly, when we use hypertonic sucrose, when we increase the amount of hypertonic sucrose in the system, we, in a concentration-dependent manner, also inhibit the activity of the PA translocase. Why is this? Well, the obvious conclusion to draw is both of these materials are inhibiting ILB fusion. Well, okay, that's quite obvious when you think of the hypertonic sucrose, which is going to be stressing the membrane because you have increased osmotic pressure, and inserting a PA molecule into this would stress it further. You'd expect that if indeed PA was bursting membrane, you would see an exacerbated effect here instead of an inhibition effect when you add more sucrose. So what about a PA inhibitor? How does this function? Would that actually inhibit the translocation of LFN-DTA? Would it inhibit the translocation of our LFN nucleic acid binding domain siRNA? Well, we tried that experiment with LFN-DTA, and I can't actually reveal the identity of these two PA-specific inhibitors. But as you can see here, when added in a temporally dislocated way, or both at once, we have inhibition of LFN-DTA moving over the PA pore. Can we quantify the delivery of cargo? Well, we radio-labeled our siRNA with 32P, and yes, we can indeed quantify the delivery of 32P siRNA into the cytosolic compartment after subcellular fractionation. Now, please don't be fooled by these grey bars, which is just oligo, because if you look at the graph on the left, you can see there's actually much less cell-associated radioactivity with the control, which and it detracts from the delivery into the cytosol, which you can also see in a concentration, in an siRNA concentration dependent way. And you can also see that there is siRNA activity associated with these events. We looked at endopore in relation to oligofectamine and lipofectamine 2000 and repeated the subcellular fractionation. And as you can see, the endopore is still functioning very well in relation to these two competitors. And we measured siRNA activity by RT-qPCR as well as by immunoblotting. Okay, so endopore was designed not only to be non-destructive in terms of enzymes, but also to not suffer from a lot of the problems that specifically charged polymers like polylysine, polyethylene suffer with regards to body distribution. So, Vitaka have funded these experiments, and we've looked at body distribution. So we made PA83, and this was labeled with a zirconium radial label, and we looked at body distribution in A459 tumor-bearing athymic mice at a range of times after intratumoral injection. After the end of the experiment, the mice were killed and various organs were removed and analyzed by gamma counting. And this is a schematic representation of the chemistry associated with zirconium labeling. Zirconium is one of the traces of choice for doing PET analysis and PET imaging. So method, just to reiterate, we grew tumors subcutaneously in athymic mice, and then the radial labeled PA part of the endopore system was introduced into these tumors by intratumoral injection, and then at various time points the mice were imaged, and then at the end of the experiment the mice were killed, various organs examined by gamma counting. So this is a more experimental design. You can see the different groups that we used and examined, and we made sure that the mice were relatively healthy and that the tumor growth was as would be expected, and indeed the tumors were palpable. This is the imaging from here. You can see the zirconium only control, and you can see the tumor lighting up. There's a bit of label there. That's exactly what we would expect. But contrast this with the endopore system on the right. You can see much hotter tumor labeling and the radionucleotide coming out via the kidneys. Of note is that there is very little or no labeling of the liver, which I think is particularly poignant. So we can do a semi-quantitative analysis of this tumor retention. You can see that here, and the tumor retention in relation to other organs. So to conclude, we have endopore cytosolic translocation. We saw that that was PA dependent and indeed required the formation of intraluminal vesicles, as the literature would predict. We saw endopore retained within A459 tumors 96 hours post-intratumoral administration, and endopore excretion was via the kidneys, and liver accumulation was not noted. And with that, I will say thank you to people, particularly my co-author, Benita, from Vitaka Therapeutics, who's responsible for all of the in vivo data. And I will thank you, the organizers and the audience, for your attention.